Before you start the lab this week, I want to give you some background information on photosynthesis and how it works. Some of you may or may not have gone over this topic already in your lecture. I know in my lecture we will not go over this until next week, so I want to give you some ideas of how it is before you actually start. So photosynthesis. This is the process of capturing light energy and transferring it into chemical energy that is found in sugars, carbohydrates. So basically you're taking the kinetic energy of light and changing it into the potential energy of the bonds in carbohydrates. And when we look at the summary of this reaction, this equation of photosynthesis, we have the substrates here on the left and the products over here on the right. Now, there are lots of intermediate molecules that are taking place from the beginning to the end, but this is the basic products over here and the substrates over here. The substrates are 6 carbon dioxides and 6 waters. With the help of light energy, they are eventually changed into a single sugar and 6 oxygens. If you know the equation of cellular respiration, you'll know that this is the same equation, but just this arrow is pointing the other direction, and instead of light, it's actually a little bit of ATP is needed and gives off a bunch of ATP. So energy is needed to have this equation going in this direction. A light energy is needed, whereas in cellular respiration, the ATP energy is being released. So this right here is an endergonic reaction, whereas cellular respiration will be an exergonic reaction, the release of energy. And this is going to occur in the in the chloroplast of plants. It'll be taking place in 2 stages. I'll go over that in a slide in the next couple slides. So again, this reaction is seen up at the top. This is a very important reaction. So when we talk about photosynthesis, this is one of the most important parts. You need to know the substrates that are going to start the reaction, the need of sunlight, and then the products that are going to be created from photosynthesis. 6 carbon dioxides plus 6 water with the energy of sunlight will help create 1 sugar and 6 oxygens. And again, this is taking place in the chloroplast. The chloroplast is a membrane-bound organelle found in plant cells. It is also found in some protistas and some bacteria, even though they do not have membrane-bound organelles. They do have the ability to do photosynthesis much like chloroplast itself. But the chloroplast is a organelle of an eukaryotic cell, and so the parts of this chloroplast, you have the outer membrane, you have the inner membrane, the space that's in between, and then on the inside of this inner membrane is filled with this fluid. The stroma is this fluid. Much like the cytoplasm of a cell, it's mostly water, but there are some other molecules that are floating around that's different than cytoplasm, but it is basically the fluid that fills this chloroplast up. And then you've got these other inner sacs, inner membrane sacs. A thylakoid is this single disc. It's like a coin shape. It is a hollow disc, so it is a thin sac of its own, the thylakoid. If you have a stack of thylakoids, it's called a granum. So it's kind of like a stack of coins will be the granum. The single coin itself are thylakoids. And then the thylakoid lumen is this liquid that is found in the inner part of the thylakoid. So how does photosynthesis work? There are two parts to photosynthesis, and it's basically the light-dependent reactions and the light-independent reactions. These are also called the dark reactions sometimes, the Calvin cycle, the Benson cycle, the carbon cycle, not the Benson cycle, the carbon cycle. And this is the two parts of photosynthesis. The light-dependent reaction, as it states, depends on light being present. So you need sunlight for these reactions to take place. They're going to take place in the thylakoid membranes. And basically with the help of light and water, you're going to, well, the help of light, the energy of light, these thylakoid membranes are going to be able to break down a water molecule, producing oxygen, but ripping off some electrons and holding on to them to create ATP and some other intermediate molecules. So in this direction here, ATP and this NADPH, which we'll talk about more in lecture, these molecules are created by ripping the electrons from your water molecule. In the light-independent reaction, so it does not need sunlight to do what it does, the Calvin cycle, the carbon fixation cycle, this is where the cell in the stroma is going to use the carbon dioxide to go through this Calvin cycle to create sugars. So this GA3P, this is a subset of a sugar. There's another picture that I'll show that kind of will have a breakdown here. And then these molecules here are giving off energy to create this sugar from the carbon dioxide. So it's basically taking the carbons from the carbon dioxide, the carbons and the oxygens, and the hydrogens that are being taken over here from the water to create these sugars. And then these intermediate molecules, they bring the electrons and hydrogen ions, dump off, and then they go back to the thylakoid to be reused in the light-dependent reaction. And so we've got a cycle of these intermediate molecules plus the light-dependent and the light-independent reactions. The same picture, or I should say the same two stages, just a different picture showing. This is taking place here in a mesophyll cell with chloroplasts. This is the chloroplast here. And so with the help of light and water, the light reactions are able to take electrons away from water molecules, storing those electrons in ATP, storing those hydrogen ions into NADPH. When you take the electron and the hydrogens away, you are left with just pure oxygen. And so that is a product of this reaction. The water is broken down and oxygen is released. Then you've got the carbon reactions over here, and this is where carbon dioxide is being brought into the cell. The carbon dioxide is being broken down utilizing the carbons, the oxygens, and the hydrogens over here from the water to create a sugar. also with the help of the ATP that is being broken down over here. So with the help of these intermediates and carbon dioxide, we go through this cycle to create sugar. And these are the final products. These are the substrates of the reaction. And all of this kind of situation is these intermediate molecules that are being created and changed and utilized to make the products. So again, light reactions, light energy is captured and converted into chemical energy. And that chemical energy is ATP. That ATP is then used to help produce sugar. Carbon reactions, or the Calvin-Benson cycle, energy over here that is created. The energy is used to produce a sugar molecule. That sugar molecule will then be stored, saved, utilized later when it needs to be broken down and used for energy. Now, we are not going over cellular respiration in this lab, but we will in your lecture. Those sugars can then be broken down by the mitochondria for cellular respiration to create lots of ATP. Kind of all of these ATP that have been created in this light reaction, the energy from these ATP is being stored into the sugar. Later, the sugar can be broken down and ATP can be created. So lots of ATP go into creating the sugar. Lots of ATP can come out of that sugar. In this light-dependent reaction, so the reaction that is involving the sunlight, it is possible by pigment molecules. These large molecules that are found inside the thylakoid membrane. These molecules are able to capture the sunlight, get excited, chemical processes taking place in the molecule, getting excited and being able to rip off the electron from the water molecule. And so these pigment molecules are the basic the major player here that is allowing photosynthesis to take place. And so there are lots of different types of pigment molecules, and we'll go over them. This is kind of the basis of our lab experiment tonight, looking at some of these pigment molecules. The different pigment molecules are found in the thylakoid membrane. Much like a cell membrane, there are different large proteins and other big molecules that are found in this membrane, and these pigment molecules are found here. So pigment molecules are embedded in the thylakoid membrane. There are lots of different types of photosynthetic pigments. We're going to look at a few. The first ones that we'll look at over here, these are chlorophyll A and chlorophyll B. Yes, we're just looking at one molecule, but there's a slight difference between the two. But these molecules are the main player of eukaryotic cells, the main ones found on Earth. When you think of photosynthesis, you think of chlorophyll. And these have a greenish color to them, and we'll look at why that is in just a second. Another molecule that we will not be looking at tonight, but this is the bacteriochlorophyll. And these are found in prokaryotic cells that conduct photosynthesis. So cyanobacteria are one example, but they have their own pigment molecules. And these are eukaryotic, the chlorophyll A and chlorophyll B. Carotenoids, or carotene. These molecules tend to be an orangish color, and they are also present, able to do the same thing, using the light energy and being able to rip off the electrons from the water molecule. Then you have this xanthophile. This is a molecule that has a yellowish color. The phycoerythrin molecule, this has an orangish kind of color to it. So why do these molecules have different colors? Why are there several different molecules that can be found in the same plant on the same thylakoid? Well, the reason that they're different colors is because they are utilizing different wavelengths that are coming off of the sun. And so when we look here at this graph, on this graph on the bottom, we have the Roy G. Biv, the red, orange, yellow, green, blue, violet. These are the wavelengths that are coming off of sunlight. We are able to see these visual wavelengths because when light hits an object, whatever is not being absorbed, the wavelengths that are being reflected from that object, our eyes will be able to see, and we perceive that object as the color that is reflecting off of it. The energy absorption is seen over here on the y-axis. And so when we look at these molecules, what wavelengths they are able to absorb. So when we look at chlorophyll A is able to absorb the violets and blues, not so much the greens and yellows, and then able to absorb the oranges and reds. And so because this molecule is able to absorb these particular colors, but not the greens, those greens are reflected off of it. And so that is why this molecule, the chlorophyll, has a greenish color to it, because those are the colors that are being reflected, not absorbed. The chlorophyll B is very similar. It is a lighter, different kind of green, but it is absorbing these blues and violets, and also these yellows and oranges and reds. And so it also is reflecting this greenish color. Now the carotenoids are able to absorb the violets, blues, and greens, but they are not able to absorb and reflect the yellow, oranges, and reds. And so as a so that is why the carotenoids are this orange color, and then the xanthophiles will be reflecting more of a yellowish color. And so why is there so many of these pigments found in the same thylakoid? And it's because they're able to absorb all of these different wavelengths of from the sun. If you were a plant in the tropics, maybe you do not need to worry about having carotenoids and xanthophiles, because you have direct sunlight hitting you, and just having the chlorophylls is enough to get all of the energy that you need. If you were a plant in the northern hemisphere, let's say in the tundra, the taiga, the sunlight that's hitting you is at a certain angle that's not all of the energy from that sunlight hitting you as if you were at the tropics. And so you might need more of these other pigments in order to capture all of the wavelengths of light so that you can get the most efficient usage of the sunlight that's hitting you. When trees start to change color, deciduous trees start to change color in the fall, the reason that is happening is because the chlorophylls are starting to break down. The chlorophylls, chlorophyll A, chlorophyll B, are starting to break down in these thylakoid membranes. And so they're not reflecting green anymore. But the carotenoids, the xanthophiles, these molecules are still there and they're still able to reflect their colors. Certain plants will, be a more vibrant red. Some of them will be more of a greenish or a yellowish color. And that's because of the specific pigments that are found in their thylakoid membranes. And this is kind of what we're going to be looking at tonight. We're going to be using some strips to kind of absorb and run some of these pigments up the strip, kind of like a dipstick assay, where the fluids are absorbed into the strip and they go through, carrying the molecules with them. Some of these molecules will be able to travel further than others. Some of these pigments will be able to travel further than others. And so you're going to be able to see them in distinct lines of chlorophyll A, chlorophyll B, xanthophyle, carotenoids. And so then you can tell which of these pigments are actually found in particular plants. Most likely you'll be looking at spinach, which is green in color, and maybe a reddish, purplish lettuce, or if you have other plants with similar coloring in your yard, things like that. You'll be choosing 2 plant leaves and looking at the different pigments that are found in those leaves. I hope this helps.